We have already talked about DNA, RNA, and genes when we talked about macromolecules, but today we want to think more about how DNA is formed, replicated, and how it's used in the coding of proteins. So who is this guy? What names do you know that are associated with DNA? James Watson, some of you may recognize his name, discovered the structure of DNA in 1953 along with Crick and a number of others. He is still alive and he's been out of the limelight as far as science for a long time, but still does guest appearances and so on. What we need to think about is how we actually study DNA today. It certainly is not the same way that Watson and Crick studied DNA nearly 60 years ago now. When we look at DNA today from a molecular perspective, we actually use viruses quite a bit to study DNA function. The odd part of this is that the viruses, as you know, are not alive. They're not cellular, they don't reproduce on their own. So how are we using them to actually study the function of DNA? It turns out that we can stimulate DNA to do particular things in the presence of viruses. Viruses are also used to carry particular information to the DNA and target it to initiate particular processes. So it's actually been very important in molecular biology in recent years. When we look at DNA as a genetic material, we think about DNA as a building block or an instruction book for coding proteins. We've talked about this before when we talked macromolecules, but when we look at the idea that originally we thought that proteins were the instruction book and that DNA, we had no idea what it did. So it wasn't until the 1940s that DNA as the genetic material or the instruction book was a much more common theme. Today we wouldn't question it, but the idea that everything about you is coded in 4 single nucleotides of A, C, T, and G is something pretty amazing. The nucleotide itself you should recognize from macromolecules. We have a nitrogenous base, a 5-carbon sugar, and a phosphate group. We look at the sugar and phosphate as being part of the backbone, and then the nitrogenous base hangs off of that. When we put all of these nucleotides together, we get a nucleic acid. The nucleic acid or the polynucleotide is what we focus on as far as DNA, RNA, and as we mentioned before, nucleotides are also used to build ATP, your cellular energy. The DNA nucleotides themselves have different shapes. They are structurally slightly different, and this means that the way they are going to arrange themselves is going to be different. Note that we have 2 groups, the purines that are A and G, and the pyrimidines that are C and T. The major structural difference, of course, being the 2 carbon rings with A and G and the single carbon ring with T and C. And this would be the same. Remember, we have U in RNA. That would be the same basic structure as a thymine, only slightly different. When we put these together, we have a repeating sugar phosphate backbone. And we look at the phosphate group attached to the sugar, to the phosphate, to the sugar, and so on. And with each one of those, you see sticking into the middle is a nitrogenous base. And when we look at putting this together, this deoxyribose sugar is specific to DNA. The ribose sugar is specific to RNA. So it's deoxyribose nucleic acid as its formal name, or ribonucleic acid for RNA. And when we put these together in its larger structure with RNA, here's the ribose sugar for RNA. We mentioned before that RNA has a U for uracil instead of the T for thymine. Fine. Other than that, structurally very, very similar. The only other thing we've mentioned before is that RNA tends to be single-stranded. It doesn't have to be, but it tends to be. So this is what RNA looks like compared to DNA. Single-stranded versus double-stranded, but we still have a sugar phosphate backbone in both cases, and we still have bases that are pointing, in the case of DNA, towards the middle, and are going to attach to each other with hydrogen bonds. And with our RNA, of course, the nucleotides are free and open. This is important later on when we start thinking about the process of protein production. Some of you may also know that Watson and Crick worked with a woman named Rosalind Franklin, and they also worked with Maurice Wilkins in determining DNA's double-stranded helix. And this is actually a picture of the X-ray crystallography structure that Rosalind took to determine that DNA is double-stranded. Now, from this picture, some of you may not be able to understand how we get DNA is double-stranded in this case. But you need to think about this like looking down from the top of a spiral stairwell. So if you're looking down the DNA, it's circling and circling and circling. And what you're seeing is through the middle, these would actually be the hydrogen bonds between the bases. So her work was critical in determining that DNA was actually indeed double-stranded. So from there, they have built all kinds of theories and made all sorts of observations about DNA. In particular, Chargaff did a lot of work on DNA composition. So looking at the numbers of A's, T's, C's, and G's. And what he found was the percentage of A equals the percentage of T, and the percentage of C equals the percentage of G. So what does that mean for you about the DNA structure? Well, it should mean to you that A pairs with T and C pairs with G. So when this double helix first came out, thinking about A pairing to T and C pairing to G was something relatively novel, of course. But when we look at the chemical structures, it makes a lot of sense. A and T actually form 2 hydrogen bonds. C and G form 3 hydrogen bonds between them. And when we look at the structure itself, you can see that. So C's and G's forming the 3 hydrogen bonds, A's and T's forming 2, and it's based solely on their chemical structure and how they come together. Now, these hydrogen bonds we've talked about before are not super strong, but they are certainly strong enough to hold together your DNA. We don't want them to be like covalent bonds. We're going to need them to break apart when we do DNA replication. So their weakness is to our benefit in the long run.